Good morning, my name is Mantasha Ahmed, and today we will talk about molecular biology and physics. Molecular biology is concerned with the study of molecular nature of basic life phenomena, variability, variation, and how this works on in generation and generation. These phenomena are these phenomena are determine the structure and properties of nucleic acid, which is informational macromolecule. It is the genes. Gene, what we know about gene? Gene is a part of DNA molecule. Okay, there is a whole stretch of length of DNA, and gene is part of it. So we know that in 1944, every McLeod-McCarthy, they did the discovery for transformation of bacteria. So molecular biology seeks an explanation for the biological phenomena in the chemistry and molecular physics. Okay, molecular physics, it deals with a wide range of life processes including enzymes, catalysis, membrane transport, mechanochemical phenomena, etc., in distinction to classical biochemistry. If we talk about that, we will talk about the structure of nucleic acid. Okay, structure of nucleic acid, the relationship. We know the structure of molecules and its functional properties, how they are interlinked. We know they in a DNA molecule. If we talk about nucleic acid, DNA and RNA, so DNA is the main molecule. We know in that the situation is different with nucleic acid, in particular with DNA. It is highly coiled. Why it is highly coiled? Because there are force of attractions like hydrogen bonds, van der Waals forces, and other interaction. Covalent bonds, so strong covalent bond, double bonded, triple bonded are already present, which provide a very good force to keep it intact. The secondary structure of DNA was established by X-ray diffraction analysis in the works of Franklin, Crick, Watson, and Wilkins. The oriented fibers of lithium salts of native DNA gave X-ray diffraction patterns containing up to 100 reflections. Now, this was the method how DNA molecule was discovered. The cross-shaped array of reflections directly shows that the structure is helical. The helical structure. Native, as you can see here, as you can see here, this is whole structure about it. We know the distance between the two strands is shown, and how the whole length of the DNA, what is it. The DNA, native DNA, is built up in a form of double helix, which is composed of two intertwined polynucleotide chains, like as you see there, whose nitrogen bases are bound in pairs of hydrogen bond. Thymine is always paired with the adenine, and guanine is always paired with the thymine, with cytosine. These pairs, Watson-Crick pairs, these are also called Watson-Crick pair, are also shown systematically here. And the molecular model of the double helix, the beta form, what we call, there are different forms, alpha, beta, gamma, and delta forms of the DNA, but what we usually follow in normal life is beta form of the DNA. Thus, the two chains of the DNA in the double helix are complementary to each other. That is, there exists a unique correspondence between two nucleotides. T is contemporary to A and G is contemporary to C. This complementarity discloses the meaning of Chargaff's rule, where the number of bonds of A bonded to T and number of bonds equal to G and C are equal. It should be 1. It should come out as 1. DNA can crystallize in various double-stranded forms at low relative humidity, like up to 70% humidity. DNA crystallizes in a monoclinic A form, that is alpha form, with the lattice parameters being as follows. A, that is the bond length, 4.04 nanometer, B 2.203 nanometer, and beta is which is 97.1 degree. Now, if we put this DNA into sodium salt of DNA are shown, the C form, that is C form of the DNA, can be discovered later. List B and C forms of the DNA They differ in pitch of double helix and angle of rotation, the rotation at which these are rotated, the base pairs. The angles of rotation between neighboring base pairs and the slope of the plane of base pairs to the helix axis. The conformational of the polynucleotide chain is determined by a set of of rotation as shown here. The angles are counted off as follows. If we look in the direction as you see here of the bond about which rotation takes place, the farthest bond like this, the farthest bond is rotated in the clockwise direction relative to the nearest one. Counting is made from this position, or as you see in this table you can see the counting and how the rotation changes by slight difference in the humidity. The conformation of this polynucleotide chain depends on conformation of the ribose sugar or deoxyribose, which we call the sugar of DNA. The sugar is present in four conformational which differ in which of the atom of the five membered C2 or C4 is brought about to the plane of the other four atoms. In the atom C2 is on the same side of the plane. You will understand from this structure as you can see. Now, if we see in this photo, the C2 is endo, C3 is endo, C2 is exo, and C3 is also exoform. The first two forms are encountered and more frequently. If we do theoretical calculation and conformations of polynucleotide chains here, and double helices are not simple. These double helices are not simple since these conformational depend on many angles. Many angles. The ribose or deoxyribose group is also conformationally is labile, what we call, according to calculations are carried out with a rigid and soft sugar. If we take that's all. Know that molecular biology seeks an explanation for the biological phenomena in chemistry, molecular biophysics. It deals with a wide range of life processes, including enzyme, catalysis, membrane transport, mechanochemical phenomena, etc. The distinction to classical biochemistry, molecular biology combines with physics, and it specifically consists exactly in the physical aspects of investigations and problems. Like we know how problems of molecular biology, biophysics are connected with the structure, functionality of the protein, and now in how in turn these protein work, protein problems. Now, like if there is some physical problem related to structure and to of the nucleic acid, so these there are mechanism in the body, there are mechanism in the genetic material which cope up with any defect that is present in proteins or genetic material. Like I can state the secondary and tertiary structure of all kinds of nucleic acid, the relation between structure and biological properties. Okay, if there is a defective protein, then a defective enzyme will be made and it will be seen like metabolism is not acting, so it will be seen from the physical appearance. It can be seen the genetic code which determine the relationship between the nucleotide sequence in the genetic DNA and RNA and in messenger RNA, mRNA, and the amino acid sequence in the protein chain. The problem of the genetic code which arises in the atomic molecule treatment of the process of protein biosynthesis in physical nature, the problem has been solved. Like if there is some sort of problem, then there are like cutters, enzymes, and deletion and adding a substrate or deleting a substrate. These are the mechanisms which are present in the system already in an organism in the nucleic acid. So any problem with the genetic material or protein synthesis, it will be solved automatically. The physical meaning and origin of the genetic code. The code is not accidental, presumably it is originated in course of uh, what we say internally pre-biological evolution. Physics is called upon to disclose the internal relation of the course and to build a theoretical model of its evolution. If there is some conformational properties of nucleic acid, the behaviors of nucleic acid like that of protein is monitored by electronic conformational interaction, theoretical and experimental investigation of the conformational nucleic acid and their changes unravel the mechanism of molecular biological process. This refers in particular the denaturation and renaturation of the nucleic acid. Like all enzymes, as I told, are already present in the biological system. Now, if we talk about, there are we have developed and there is evolution, so much evolution. So we have developed process pre-biological, biological evolution, ontogenesis and carcinogenesis, which lean on to known properties of nucleic acid. There is a list to claim to be complete. Some of the problems mentioned are already, which I mentioned, have been solved. Other are still in initial stage of resolution. So, if we want to know